Welcome you all, myself Pransu Kumar Pathak. And dear friend, I'm really very happy today to share an information with you that I got first publication in my life that is that is really means a lot for me. That is my first paper of my life, and I would like to thank all my colleagues, especially Rahul Michael sir. He really guided me a lot in my starting of my PhD. I'm really glad to discuss about my group. I'm really happy with my group, and each and everything of life is really very glorious and very fabulous. So dear friend, try to enjoy your life and try to be focused on science, and science is really very beautiful. So with that note, I will progress and I will go through my lecture. So dear friend, try to be very punctual and try to be very informative. बहुत जब तक आप informative नहीं बनोगे, selective नहीं बनोगे, तो you will really suffer in your life. So I would like to again thank each and every member of my group, and thank you so much for your support and kindness. For stay tuned with us, and especially this particular lecture is focusing on plant immune system. We will discuss about plant immune system. So dear friend, I would like to first demonstrate about what is serial dilution. Okay, so dear friend, to know about serial dilution, you have to understand the basic things. The basic thing is, try to go in your field and try to take a rhizospheric soil. What is rhizospheric soil? When a plant is planted and just beneath the soil region, root region soils, if you will take soil from this region, you have lot of microbiota. Microorganisms are a lot. So what you will do, when you will take 1 gram of that soil in 10 ml, that is called this stock preparation. So now you can do what? This is 10 ml of total. So when the serial dilution means, you will take 1 ml of that solution, that is a suspension solution. Actually this is suspension and colloidal or true solution. I am not going to discuss a lot about this. Just I want to tell you, whenever you will take 1 ml of that solution and you will pour milky water 9 ml, then again it will comes to 10. The colony will decrease gradually, and there is a time where there is no colony will grow on the plate. So this is called serial dilution. So serial dilution is very important for plant pathology student. Basically those students who are working on plant microbe interaction, so they used to do with the samples. So this note, I will thank you again. And dear friend, we have to understand, there are a lot of stresses are reported in plants. So plant immune system is much more complex than animal. So I will discuss it a lot, but biotic stresses, abiotic stresses, but this topic of discussion will focus on biotic stress. Okay, so biotic stress is really very important to understand, guys. Okay, so plant pathogen and interaction and agents really very devastating. So up to 30% in normal annually loss is due to the plant pathogens. It may be bacteria, virus, or anything. I will just upload my previous lecture link that will help you to correlate this topic. Okay, so the loss of the crop up to a 30% maybe in a normal case. Or Xanthomonas oryzae, this field is caused by Xanthomonas oryzae pathovariety oryzae, that is Jew, and Jew can loss up to 80% or 90% in the case of severe attack. So this is a normal case. ये आपको अक्सर दिखता होगा फील्ड में. तो this is nothing. This is a bacterial blight disease which I have discussed in the subsequent lectures. I have discussed a lot about this. So go there and try to learn about bacterial blight and all the diseases, okay, in plants. So here the topic of discussion is plant system have immune system too. हमारे तरह जो immune system, जैसे हमारे यहाँ blood circulation है, हमें blood cells हैं, immune cells हैं, but plant doesn't have this complex creation. But plant immune system is much more efficient in comparison to the animal. Okay, so the question is, whenever plant challenge, the question is, whenever plant challenge with any pathogen, then how they respond to particular pathogen? That's the first question. Now the second question, as like animal, plant have any defense mechanism to combat with this pathogen? और अगर है कोई कॉम्बिनेशन, है कोई ऐसा प्लांट पैथोजन इंटरेक्शन, तो कैसे वो एक दूसरे को अपार्ट करते हैं, कैसे एक दूसरे से रिस्पॉन्ड करते हैं? तो रिस्पॉन्स and interaction is really very important to understand. Okay, so in this particular lecture I will go through detail about these things. Okay, what they do and how they protect themselves from these pathogens. So the basic plant immune system. Before going to in deep, I would like to say only one thing. As we start with immune system, first thing in my mind comes that blood circulation, blood cells, immune system, antigen, antibody, interactions as like animal. Nothing is possible in the plants because there is no circulatory system as like animal. Blood is not there, immune cells is not there, है ना? अभी तक रिपोर्टेड नहीं है कि प्लांट्स में कौन कौन से इम्यून सेल्स होते हैं. एंटीजन एंटीबॉडी इंटरेक्शन इज नॉट नोन इन प्लांट. सो बट unfortunately plant doesn't have this complex system for immunity. But dear friends, plant have strong immunity than any other animals like human also. So हम लोगों से भी ज़्यादा अगर आप जब देखोगे तो plants are more having immune system. So this is really very important to know. So lot to know, lot to discuss. Come to the next slide. Dear friend, there are plants pathogens basically characterize. I will characterize them. Fusarium belt. So fusarium is a corn disease that is caused by fungus, and root knot disease caused by nematode, which I have already discussed. This is bacterial disease, bacterial sheath blight. Okay, sheath blight is different than bacterial blight, so don't confuse. This is called rice blast. So blast is caused by fungus again. Fungus. As you can, soybean yellow mosaic virus. So this is virus. So अगर bacterial blight की बात कर लें, bacterial leaf blight (BLB). So bacterial leaf blight is bacterial disease, fungal disease, nematode disease, viral disease. So these all pathogens are what they are doing. They are, they are disturbing plant system, and plant is responding to them. So understanding this mechanism is really very important, you guys. Okay, so let's come together and we will try to learn what fungus have their intrinsic property. So fungus have a special characteristics. Then fungus start feeding from the host by formation of host area. What they do, fungus can enter into the plant cell through plasma membrane and try to connect them. So after connecting, they are feeding. They can feed on that particular host. So hyphae is inserted in a form of host area, and then fungus get nutrients and all the content from plants in that particular organism, that particular plants. So this is the way the fungus can disturb the plant immune system. So now the bacteria. I have already told you that bacteria cannot cross this barrier that is called plasma membrane. So if plasma membrane is not crossed by this pathogen, how pathogen will interact with the plant cell? So dear friend, plant cell have maximum receptors. Receptors are bonded to the plasma membrane. These pathogens start secreting toxins, and these toxins have capacity to interact with these receptors, and this receptor internalize them into the plant cell. So again, what bacteria is doing? They are secreting toxins, and this toxin is going into the plant cell and disturbing their complex system. So this is the action mechanism of bacteria. Now come to the virus. What the virus do? Virus enters into the plant directly, and virus can easily go and easily pass through plasma membrane. As like bacteria cannot do so, but virus can enter into the plant cell and modulates the host system and host metabolism, and hijack the system, hijack the plant cell, and try to what they are doing. They are transcribing their own RNA, protein and all, and they can collapse the plant cell. And in this way, plant is dead. So this is again very interesting story to know, guys. So this is again very important story, and try to know, try to focus on these particular topics. So now come to the next. I am discussing about Pseudomonas syringae. Pseudomonas syringae is a well-known disease of Arabidopsis thaliana. Arabidopsis thaliana is a model plant of organism of plant system. So I am also working on Arabidopsis thaliana. I have already I have published a paper with our group. So I just want to tell you that Arabidopsis thaliana is really very interesting, and I am working on mutants. Mutants play a lot. Mutants को grow ही करना बहुत मुश्किल है. बहुत सारे ऐसे mutants हैं you cannot grow them. If you will grow, they cannot survive. So seed collection is very tedious job, and I am facing a lot of problem in this also. So it is my story. I will discuss another time. We will discuss first. So as like plant pathogen except virus do not enter the cell wall. अब सेल के अंदर प्लांट्स वायरस के अलावा और कोई इंटर नहीं कर सकता. ये क्या कर सकता है, Pseudomonas syringae क्या करता है, एपिडर्मल सेल लेयर्स में घूम रहा होता है और वहां से टॉक्सिन प्रोड्यूस करके क्या करता है, पौधों के अंदर चला जाता है. सो दिस वे bacteria or viruses, bacteria can not enter into the cell directly, but it can interact and can produce toxins. Now come to the root knot disease, that is called by nematodes. Actually, nematode have a special characteristics. They can enter into the plant host system through the stylate. What they do, they form a stylate formation, and this stylate enters into the host organism, and by the penetration into the plant cell, a stylate is a feeding organelle. Now this stylate is used by nematode, and nematode can suck all the nutrients and plant can get affected. So this is again very important story to know. So lot to know, lot to discuss. There are lot of diseases. I cannot go through each and every disease, but I just want to tell you one thing, that these things are really very important to know. So try to focus and try to understand how these pathogens basically works. So these are the model how these bacteria, virus, nematodes are working into the plant cells. So now come to the story. Defense response in plant, maybe it's of a structural defense, maybe it's chemical defense, or the defense mechanism may be constitutive, as you can see here. Okay, so it can be constitutive and it can be inducible. So inducible means you are responsible for that. You will induce that disease and you will try to know the defense mechanism of plant. So this is really very interesting topic of plant pathologists. Pathologists always try to introduce some disease and try to cause postulates. If you have time, go onto the Google and try to know about cause postulate. Cause postulate कहता क्या है कि if you are introducing a disease into a plant, it will appear, but if you can cure that disease by removing that particular compound, or you can treat them and treat well to remove that particular complex compound. So this is inducible nature of defense response. Now a constitutive, plant have innate immune system and an inherent property to combat the pathogen. So some of the genes are basically inherent property of the plant that can constitutively work. Constitutive means लगातार हमेशा defense response plant के अंदर generate कर रहा होता है. Now how these microorganisms, bacteria or virus or any pathogens respond. Sometimes plant produce peptide antibiotics. So by these antibiotics, bacteria can kill, or there is a competition between plant and pathogen. So who will win? This is again a race. Antibiosis and food quenching is really very important. Food quenching, I just want to explain briefly, because during my MSc, I used to do, in my dissertation, I used to drop a fungus and try to, give a bacterial combination. So this bacteria and this fungus fungal growth is restricted due to the bacteria. So bacteria have that property. So by extracting that metabolite from that bacteria, I can manage this fungus. So fungal disease can be managed through bacteria. Through what? Competition. No, this fungus and bacteria is competing for the life. Who will strong? They will win. So this is the story. So competition, antibiosis, peptide antibiotic, food quenching. अब मान लो कि बैक्टीरिया ने सारा फूड क्या कर लिया, ऑब्जर्व कर लिया. अब फंगस के लिए कोई फूड सोर्स दिख नहीं रहा. वाई? बिकॉज बैक्टीरिया ने क्या किया, हाईजेक कर लिया पूरा न्यूट्रिशन सिस्टम को. अब फंगस जो वहां पे है इज स्टार्विंग, एंड ड्यू टू स्टार्वेशन अल्टीमेटली फंगल विल डाई. Fungus will die. So this is the story of fungus starvation, and this technique is really very useful in management of fungal disease. Now come to the structural defense response. So what plant can respond and how can structural defense response is generated into the plant? Cell wall alteration, cell wall structure will change, epidermal cuticle wax deposition. Sometime plant to protect from these pathogens, what plant they did? They do wax deposition on the cuticle surface. So callus deposition, lignification, stomatal closure. The physical means of transport of this pathogen is through this stomata. So if stomata is closed, so how can pathogen enter? So this is again a very challenging task. So what I am showing here, that plant stomata is closed. So if plant stomata is closed, pathogen cannot enter. And in this case, if it is open, pathogen can easily enter into the cell and enter into the plant system and can, what they did? They can invade and create a bonding tissue and enter into the tissue and infect the whole plant. So this is really very interesting topic to understand, guys. So now come to the chemical defense. Plants have special characteristics to respond about this pathogen by secreting such compounds that are produced into the plant. This is called as secondary metabolites, on which we are working. Our topic of research is metabolomics, and our whole group is working on metabolite pathway engineering. So in our lab, we are all of us is working on terpenoid pathway or flavonoid pathway or so and so forth, so many pathways. So we are working on secondary metabolomics, and we are trying to alter the gene expression, and I will our main focus of our lab is to pathway engineering, and pathway engineering is really very important job. So this is really very important task. So terpenoid production, phenolic production, alkaloid production, and proteins, some like chitinase, glucanase, and defensine. These all things can produce chemical defense, and this defense mechanism can help plant to combat that particular pathogen. So this is again very important for you guys. Try to understand. This is really very important slide for you. Let's come to the what they do, how they protect themselves from the pathogen. So this I have given this thing because there same thing is happened with us also. कभी क्या होता है, इन्फेक्शन होता है. अभी जैसे कोविड-19 हो गया, तो इन कोविड-19 के दरमियान जो हम लोगों में इम्यून रिस्पांस जनरेट हो रहा है और जो ठीक हो जा रहा है, तो प्लाज्मा थेरेपी का भी नाम आपने सुना होगा. व्हाट प्लाज्मा थेरेपी मीन्स? बिकॉज देयर सेल नो अबाउट कोविड-19. So if that particular pathogen again enters into the body, then it will generate response. That is called immune response. So infection that will leads to recognition, and recognition will try to inform that plant by local resistance. This is called induced systemic resistance. And another thing, जब local response से भी काम नहीं चलता है, तो plant would like to respond in a more specific way, that is called systemic acquired resistance. So systemic acquired resistance is very specific type of resistance, acquired resistance. Okay, so this is systemic, means the whole plant में, and local resistance means in a particular region of the plant. Okay, so local resistance I will discuss, and systemic acquired resistance I will also discuss, but try to note down these steps. Now, after infection and reinfection, or suppose reinfection is going on with the same pathogen, so what plant will do? Plant will develop memory. So this memory can help to formation of origin, which I will discuss. In plant, basically origin development is always through the subsequent generation after generation infection of that by the same pathogen. Plant have a special characteristic to develop origin into the body. So this plant can have special characteristic to combat with that pathogen, that is called an origin, which I will discuss in subsequent slides. So dear friend, important causative agent and all I have discussed a lot. So I have taken again these things to make you understand. So I will not discuss a lot about that, because I have discussed. I will put link in my description box. If you are not known about these things, so try to go on my previous lecture series and you can understand from there. Okay, and in my description link I will give you the link about these things. And in basic funda, I will just tell you one thing. Biotrophs eat to kill and necrotroph kill to eat. So in a brief way, I will just describe about necrotroph. So necrotroph, what they do? Necrotroph kill the cell and then consume the content of the host organism. It means in necrotrophic organism, plant or that particular host is dead, and on their dead tissues this pathogen can interact and they can survive. But in the case of biotropic, life within the host organism without causing death. If death अगर हो जाता है host का, तो ultimately pathogen cannot survive. So in this case, pathogen also will die. So biotropic pathogen have only capacity to interact with those living tissues only. If pathogen will die, if plant will die. But the most important and most ambiguous is hemibiotrope. Hemibiotrope, it is switch from biotropic to necrotroph. पहले तो बायोट्रॉपिक नेचर शो करेगा और फिर नेक्रोट्रॉप में भी रहेगा, मतलब डेड हो जाने के बाद भी वो क्या है, सिस्टम में रहेगा. So this is the nature of hemibiotrope. Some type of person is also of this nature. So be protective from this type of person. आपके लाइफ में भी ऐसे अगर लोग हैं तो उनसे दूरी बना के रखें, ज़्यादा अच्छा होगा आपके लाइफ के लिए. Okay, so come to the next slide. I just want to tell you how biotechnology concepts comes in plant immune system. So this paper published in 2010 in Current Opinion of Biotechnology, and in this paper authors try to know and try to give you information that how classical breeding strategy is not have a problem of linkage drag. So I just want to tell you what is linkage drag. Suppose your gene of interest is this. Okay, so gene of interest is this, but from that gene contains some other gene, and this gene is very closely related to that gene of interest. So what will the consequence of that? So the consequence is all this system, all the gene frame will go into the plant. अब सोचो, if whole thing will go into the plant, what will be the cause? What will the consequence? The consequence is linkage drag. Linkage drag का मतलब क्या? Undesirable trait into the plants. आप उसको desire नहीं था. That is not your desire. So what you will do? You will just remove these things. So removal process is always led by backcrossing. So this is a breeding program. So this is very tedious and time consuming process. पहले तो linkage drag होगा, फिर आप linkage drag को हटाओगे through back crossing. What is the probability? Probability is again 50% of removal. अगर वो linkage drag इतना मतलब close है, recombination they are closely linked. याद रखिएगा, linkage drag तभी होगा जब two genes are closely linked. कभी भी बहुत ही distantly part gene cannot be a form of linkage drag. Okay, so this story is really very important to understand, guys. Okay, so classical breeder have a problem of linkage drag. But in biotechnology, you can insert any gene from anywhere to anywhere, any place to anywhere. You can do anything in the field of biotechnology, as like BT cotton, flower, strawberry, tomato, and so on and so forth. So many advancement in the field of biotechnology happened. So, dear friend, I am discussing these things in details because I just want to not make you, I want to make you efficient. Not, पढ़ाना और लिखाना तो अलग बात है. हम आपसे बातें कर रहे हैं. I'm just giving my time to you and try to utilize this. This time is really very helpful. मैं रात में बैठ के आपके लिए लेक्चर रिकॉर्ड कर रहा हूँ. Why I'm doing this? Because I want to share my knowledge. If I cannot share this, it means a total wastage. So I can't waste my knowledge. So I'm just utilizing and just I'm conveying my message that whatever I am doing, I will try to convey to you. So try to learn. Okay. So come to the next. So in this paper, what they do in the first, improving the pathogen recognition. Pathogen recognition को कैसे हम improve कर सकते हैं? So how will you improve the pathogen recognition? So pathogen recognition can be, can be enhanced by PAM molecules. Effector recognition through plant pattern recognition receptor, PRR. If your PRR is strong or resistant protein, the R protein, trigger the plant immune response, improving microbe recognition capacity in plant, boost plant immune system pathway into their entire whole. So if this PAM molecule will, recognized by this R protein, plant have a special characteristics of memory, and thus signaling cascade is going on to boost the immune system of plant. So PAM molecule is what they are doing. They are just, thus they are triggering the immune response by improving the, microbe recognition capacity. So plant is boosting themselves. खुद को वो क्या कर रहा है? Boost कर रहा है by recognizing that particle. So this particle is really very important. Now in this second case, what they are doing? They are, intermediate component of plant immune signaling pathway improve another way. Another way. This way is right. If this pathway is blocked, plant can recognize another cascade of, cascade of reaction. And this case cascade is known as pathogenesis related protein, that is NPR1. So this is again very important for that pathologist, and जो plant pathogen interaction में काम कर रहे हैं, these things are very important for you guys. So as like Burkitt transcription factor, Burkitt transcription factor have very much important role in all type of plant processes. So this is ubiquitous, Burkitt transcription and some other transcription factor. High 5, which I have published the paper, this is also a transcription factor, light regulated transcription factor. So this is really very important to you to know, guys. So dear friend, in third part, in the C, infection, plant pathogen deliver a multitude efforts into the host plant. बार बार pathogen आएगा तो virulence pattern trigger the plant immunity, the host immunity will improve. So if novel root and novel system will develop, plant get immunized. So now the last part, that is plant pathogen priming. So what is the meaning of priming? Priming means the beneficial soil microbes, which I have already, why I have covered in first serial dilution. If you know that PGPRs, plant growth promoting rhizobacteria, are microorganism, PGPMs. So these bacteria or virus have capacity. This, that particular, जब आप rhizospheric soil लेते हैं, तो there are so many beneficial microorganisms. So if you will spray prime these plants with that particular, so by priming them, you can boost their immune system. So this is again very important story. So dear friend, plant have two type of immune system, pattern triggered immunity or effector triggered immunity, which I will discuss in lecture. Thank you so much for watching part 1. I will continue this topic in subsequent lecture. Bye. Take care. Thank you so much for watching this.